Within all of us, we have information encoded about us in a molecule called DNA. DNA, however, is really small, yet it can tell big things about us, including our gender, our height, eye color, abilities, and even our chances of cancer. But how can we read something so small to be useful? Here at the University of Texas at Austin, we have developed computational DNA mechanisms to detect certain characteristics of a DNA or RNA sequence. DNA computing works because one half of a DNA strand has nucleobases that matches with its other half, forming base pairs. When there are more matching base pairs, there's a stronger attraction to bind together. Using this concept, we want to detect a point mutation in the MDM2 gene because this mutation is known to increase the chance of cancer. We first combine the strands of the MDM2 DNA we want to test with the two DNA catalysts. After those have been bound, we add two DNA hairpins. Because the catalyst position, the hairpins can unzip with hairpin 1 attaching to both catalysts. Now hairpin 2 can also unzip, forcing itself onto hairpin 1 because of a stronger attraction. But wait, what if the base pair on our DNA was actually right and there was no mutation? The catalyst will come in and again bind to MDM2 DNA. But because the base pair is correct, the catalyst binds a little more strongly. The hairpins come in, but because the catalyst has a different position, hairpin 1 is unable to bind really well, and therefore hairpin 2 cannot bind either. So when there is a point mutation, we can get hairpin 1 to bind to hairpin 2, which we could quantify to see how many point mutations there are. In order to do this, we add a reporter complex which consists of a fluorophore and a quencher. The fluorophore molecule can be excited and release light, but because the quencher is currently attached to the fluorophore, it is suppressed. However, because of competitive binding, the hairpin complex can remove the quencher strand and bind to a fluorophore, which now can be excited much more. Therefore, by detecting the amount of light released, we can detect the amount of point mutations in a sample of MDM2 DNA strands, which can predict the chance of cancer. Using a similar mechanism to detect a point mutation, with slight modification, we can also detect an apurinic site, which is caused by ricin, a toxic protein. In short, we add the DNA in question, the catalyst, the hairpins, and the reporter complex together, and because the level of light emission corresponds to the amount of mutations, we can draw conclusions from the amount of light emitted. In the future, we hope this concept can be portable and used in the field. We would like to thank the Ellington Lab, the College of Natural Sciences at the University of Texas at Austin, and IDT DNA for the possibility of this research.